he has also contributed to our center in our policies of preventing and treatment of CMV, polyomavirus, bacterial infections, just to mention a few of his collaboration. And he now will certainly help us with our xenotransplant group. If you don't know, the University of São Paulo has created 5 years ago a xenotransplant group. We are very advanced in this area in terms of now we have implanted the embryos and we are expecting to have the new genetically modified pigs in a few months. And he will certainly help us with this new field. And finally, he's my very and good old friend. Please, Jay.
Well, thank you very much, and I apologize for my misunderstanding about the time zone differences. I think that's a function of old age or something. And I have many good friends there, and was in Brazil 15 years ago at the first one of these symposia, and I'm proud to be part of what you're doing. I've been interested for over 20 or 30 years in xenotransplantation. And what I'm going to do is give you a view, and I'm more than happy to help going forward. But the real basic idea is that we don't have enough organs for transplant. I think in addition to that, we need to know that we need to understand the biology of graft rejection better than we have done in the past, and xenotransplantation can help with that. So, what can we do instead of taking animal organs? Well, we have all of these new treatments, but you may have heard, for example, there are problems with genetic therapies. Some of those have been interrupted by individuals dying of complications of the therapies. The artificial organs are coming along, but are not ready for use yet. And so many of us are interested in xenotransplantation. This has been, of course, going since the 1600s, when they've been trying to put pig organs into, or various organs into people, but was accelerated in 2021 and 22 with the first decedent studies and then the first live recipient of a pig heart that was genetically modified.
Quatro é você ou eu
I
que...
should say that in 2001, I received the patent for cloning and sequencing the porcine endogenous retrovirus, which slowed down the field a little bit because of regulatory oversight. We use pigs because they're easy to breed. They can be of a good size match, but they have immunologic disadvantages that need to be addressed before you can use them in human subjects. There are also some metabolic incompatibilities, such as uric acid metabolism, that are a little bit different. And so we need to think about those going forward. And just to give you an example, and this slide is overly complicated, but because all humans have natural antibodies to the sugar that is present on pig endothelial cells, it starts the complement cascade. It starts the rejection process. And so that what you get is systemic clotting and coagulation, coagulopathy, as part of this early syndrome. And what you see when you put a pig organ that's unmodified into a human or a non-human primate is within minutes, the organ will shut down with hyperacute rejection. If you breed your animals without the alpha-gal sugar, you can get past that hyperacute rejection, but you still have early xenograft dysfunction, part of which is metabolic and part of which is ischemia reperfusion, which has recently been addressed by better artificial perfusion system. We then get another round of graft rejection, which is immunologic, which is antibody mediated and to some extent T-cell mediated, and then chronic rejection, as we might see in allotransplantation. As a result, there are up to 10 or 15 genes that have been modified in pigs to reduce the incidence of both immunologic rejection and complement activation, as well as inflammatory activation. But in addition to that, we put in B and T cell depletion, we do complement depletion, and the backbone of this type of transplant, as opposed to what you heard about earlier with allotransplantation and the calcineurin inhibitors, here is co-stimulatory blockade, anti-CD40 or other related agents. In addition, we do a fair amount of supportive or prophylactic antimicrobials. From the infectious disease perspective, the goal is to try to make this safe. How do we do that? What we do is we develop assays for relevant pig organisms so we can track them both in pigs and in humans. We need to learn about the biology of some of those. And I mentioned the porcine endogenous retrovirus, which I cloned about 20 years ago. And we need to understand the impact of immune suppression on these organisms if we are going to put them into humans. For this audience, for the infectious disease community taking care of immunocompromised patients, this is just what we do in allotransplantation or stem cell transplantation or other immunosuppressed hosts. Does an organism cause infection, and can we prevent it or deal with it once it's occurred? This is a little different as we go into clinical trials. Each clinical trial is, I'll use the word package, but it's basically has a number of components which are fairly straightforward. Each recipient needs a specific organ, a heart or a lung or a kidney or a liver. There are patients who have their own background infections with cytomegalovirus, Epstein-Barr virus, and the like. We have a source animal, and each source animal will have different genetic modifications that will make it more or less acceptable immunologically. And then we have all these different ways of monitoring the recipient. But each is a package of the immune suppression that we're using, the recipient and the host, just as it is for allotransplantation. Now, this is old to all of you. This is from my early papers. But basically, there is this semi-quantitative relationship between the epidemiology or exposures of the individual going into immune suppression and the immune status of the host, what we have called the net state of immune suppression. And that is basically the effect of the immune suppression plus all the other things we do to patients to make them more likely to develop infection. And that includes central venous catheters and lines and drains and surgical procedures and the like. The risks are enhanced in xenotransplantation, say compared to normal zoonotic infection from pigs, because we're putting the organ right into the individual, and we may not have the appropriate assays, the microbiologic assays, to detect those particular organisms. In addition, most people will not have pre-existing immunity to pig organisms. And you should think about COVID-19 in this regard, because that was one of the novel things about COVID, is that none of our patients had pre-existing immunity that was meaningful to that. Well, none of us have pre-existing immunity to most of the novel pig pathogens. They may cause novel clinical syndromes, and the genetic modifications we put in may alter the host response. Now, pig breeding in the United States and everywhere is heavily regulated, but mostly to make the food safe. And therefore, they exclude a certain group of organisms like salmonella just to protect us when eating those particular pigs. But the equation is different in xenotransplantation. Yes, we have common human pathogens of allotransplant recipients. So it's Epstein-Barr virus and CMV and herpes simplex and the like. And we have good assays and generally good therapies for those. There are also some traditional zoonoses, Toxoplasma gondii. We again have reasonably good assays and therapies for toxoplasma. There are species-specific organisms that we think are only infectious for pig. And that would predict that the infection that we see, if we saw any, would be restricted to the xenograft. Turns out that this is not quite accurate, and I'll go back to this in a little bit. There are some organisms like adenovirus and others which are very common and for which we may not have assays that are validated for the pig type of these organisms. And then the real fear of the community and of the medical community is of unknown pathogens, of organisms which we've just discovered. And again, that was what the porcine endogenous retrovirus was when I cloned it so long ago. And so based on those categories, I put together a list a long time ago of potential pathogens from pigs that would be important in human transplant recipients. And there are a few that are in red that we really have to deal with because they may not only cause infection of the graft, but they may cause systemic syndromes, even though they may not infect human cells. They may cause systemic syndromes that are dangerous to the human. And I would mention a couple. Swine influenza, can infect human lungs. Porcine hepatitis E virus is also a human pathogen. Porcine cytomegalovirus is an important pathogen for pigs, but will stay in only pig cells, and I'll show you the science behind this as well. And then some of the other viruses will have different clinical presentations. And so this became what I call designated pathogen-free miniature swine to protect the recipient from what we thought were the most common or likely pathogens derived from pigs that would affect humans. And over the years, there have been a number of different assays developed that are molecular assays or serologic assays. Joachim Denner uses a lot of electron microscopy, and there are quantitative PCR reactions. And so based on those lists, we now have some assays that we didn't have 20 years ago. But what I'm alluding to is that some of these will be unimportant. Some of them will be located in the graft, and some of them will cause systemic infection. But until we do clinical trials, we won't know which is which. Let me go back and just talk about a couple of these viruses in a little more detail, because you may read about them in places like the New England Journal and elsewhere. The porcine cytomegalovirus, which was the virus they found in the heart recipient at the University of Maryland, is one that we've been dealing with for a while. It can be bred out of the colony by cesarean derivation of pigs. Interestingly, even though it doesn't cause endothelial infection elsewhere in the body, it causes activation of the pig endothelium and coagulopathy. And therefore, it causes a systemic syndrome that looks a lot like sepsis. The porcine lymphotropic herpes virus doesn't infect humans, causes lymphoma in pigs. And the porcine circovirus can cause pneumonitis in pig lung tissue. I'll come back to the porcine endogenous retrovirus, but basically it's a normal virus of the genome of pigs that doesn't cause any disease in them. And just for completeness, you've just heard about SARS-CoV-2. SARS-CoV-2 does not infect pigs, and pig coronaviruses do not infect humans. The porcine cytomegalovirus infects only pig cells and tissues, and we've developed diagnostic tools. Infection looks very much like it does in human CMV, and this is a picture of the intranuclear inclusion of porcine cytomegalovirus in a ureter. You will notice that when we put this into a baboon, that viral activation occurred in the ureter and the kidney, but no infection occurred in the baboon cells of the host. But endothelial activation, seen here in brown, occurred throughout, and coagulopathy became a major concern. When PCMV was reintroduced into our pig colony, all of a sudden our survivals of xenografts were reduced by over 50%. And this is due in part to the fact that there's elevated class 2 expression and early graft rejection. So PCMV is bad from the perspective of systemic coagulopathy, but also it seems to stimulate graft rejection. Our drugs don't work very well. This is to say that in non-toxic levels, only cidofovir seems to have activity against porcine cytomegalovirus. Ganciclovir didn't work well, acyclovir didn't work, foscarnet didn't work. And cidofovir, as we know, is fairly toxic for kidneys, so it's not a very good choice. So we'd like to not have that in our breeding colony. The porcine endogenous retrovirus that I spent a number of years working on, we cloned in 1997. It varies in terms of copy number and how it appears in different pig strains, but it's pretty ubiquitous. It's pretty much present in all pigs. After we cloned that, Britain and the United States blocked all xenotransplantation in 1997, so that we had to develop alternatives at that time for clinical xenotransplantation. In the interim, we worked out the biology of this virus and showed that, in fact, PERV, porcine endogenous retrovirus, was in fact 3 viruses, A, B, and C, and then a recombinant, AC, which was more virulent, tend to replicate better than any of the other viruses. PERV A and B can infect human cells. Human cells have a receptor for PERV A and PERV B. PERV C infects only pig cells, but the recombinant infects PERV AC, infects human cells as well, but only a specialized cell line, an adenovirus transformed cell line. So this is important because there's never been PERV infection seen in any humans exposed to pig tissues. So that we think the risk is quite low, but we want to be very confident about that. So we've developed molecular assays for all the different strains of PERV. And this is the recombinant, which has the cellular binding domain from A and the replication machinery from PERV C. When we looked at this during natural infection in pigs, it turns out that AC is formed and goes back into the pig genome. So this virus is amplified in any pig that's carrying both A and C. How do we deal with that? Well, we can find animals that don't have PERV C or that don't seem to transmit the virus, or what you can start doing is genetically engineering animals that don't have PERV C. And so we showed a number of years ago that we could, using CRISPR-Cas9, knock out all the copies of PERV in a given cell line. In this case, it was 62 copies of all the PERV elements. And then they used the same strategy to develop pigs that no longer expressed PERV, knocked out the polymerase gene of all the copies. And so therefore, we were able to remove PERV from concern for clinical xenotransplantation. So again, those of you who know me know I love these timelines of post-transplant infection. And what I'm trying to convey here is that you will see common human infections because these are immunosuppressed humans exposed to the outside world. And you will also might see some pig-derived pathogens. And so what you will see early are largely technical complications in the first 2 or 4 weeks after infection. But as the effects of immune suppression accumulate and get stronger a month or so after transplantation, you not only will see things like pneumocystis and cytomegalovirus and cryptococcus, but you'll start to see the porcine viruses as well. So that if we breed them out of the colony, you won't see those. If you don't breed them out of the colony, you may start to see them. And in fact, that was the period when they saw the porcine cytomegalovirus in the recipient of a heart at the University of Maryland. So it's now a combined timeline of short-term and intermediate-term effects of immune suppression. To this end, we have a variety of microbiologic assays so that we can screen the source animals that we can monitor the xenograft recipients, whether they're sick or not, and maybe we should be monitoring hospital staff who are intensively exposed to these viruses, or who, for example, get splashed with body fluids from the pig or from the recipient. So we need to think about infection control measures. Now, we're all used to this in the era of HIV and then again in SARS-CoV-2. But we need to think about ways of monitoring the recipient, of keeping them isolated in case they're infectious for other individuals, keeping them isolated in the hospital, and to have protocols in place if they get admitted to the hospital for how we're going to deal with them from an infection control perspective. We need to decide how to handle surgical equipment that's used for these individuals and whether or not we need to monitor the surgical teams, both for the pig and for the recipient. And we need to have a plan in case they get exposed to bodily fluids. What do we do about their social and sexual contact? Well, we need to talk to them. That needs to be a part of the informed consent process when we start thinking about clinical trials in terms of whether we monitor them or not. Now, this is my favorite picture of our regulatory authorities. I'm assuming the members of the Food and Drug Administration from the United States are not listening. But how do we get to clinical trials? Each xenotransplantation trial is a package, as I've said, of an organ, a recipient, a level of immune suppression, and a specific pig. The infectious risk is not zero. It's never zero in transplantation. So we have to do what we can to reduce that risk. Part of that is careful screening of our animals. With immunosuppression, we can expect human pathogens and potentially pig pathogens to be activated. And so we need to make decisions about important pig pathogens in advance of clinical trials. We need to have an archiving system. We need to put blood samples away for testing against epidemiologic studies in the future in case something happens. One of the things I haven't mentioned is that pig cells are resistant to some common human viruses. So in addition to being able to screen these pigs in advance, as we do not do for human organs, they are also resistant to hepatitis B and C and to HIV. But we will only understand this risk when clinical trials emerge. For example, primates cannot be infected by porcine indigenous retrovirus. So all the primate studies are not useful in that regard. So we have to go into clinical trials to learn more about the risks of xenotransplantation. So we would say we need to be careful, but there is an opportunity here. Although there are risks, that we can save lives. That if we do safe pig breeding, that we monitor our patients and our staff carefully, and that we're gentle with our immune suppression, that we should be able to go forward in the field of xenotransplantation. So this is what Massachusetts General Hospital looks like. Not today, it's freezing today. And if I can help you going forward, I'd be more than happy. And thank you again to my friends for inviting me to participate in this important seminar.
Não tinha nenhum, veio suspeito do PS. Agora na enfermaria não sei o que. Dá para entrar no link aqui da gente, ver quantos Covid tem.
Elias, you're muted.
Não, no nosso aqui, que mais. É, quem tá nosso. Quem tá, a Amy tá vendo, né?
Aspect of xenotransplant that we need to face in the very near future, and we are already facing this problem. I think we should open for questions, but I have one question. Many of the epidemics that occurred in the humanity came from the relationship of man and animals, wild animals, as COVID is an example, and you have mentioned this. Do you think we are running the risk of having another epidemics by having this close contact with the xeno organs?
I don't think so, but it's a very important question. It's important both biologically, but also for the public acceptance of the clinical activity. So most of the important infections we've had have had a mechanism for getting into the human population. Certainly that was true with all the plagues and all of HIV and the coronaviruses. The concerns about porcine endogenous retrovirus, for example, therefore are realistic. And so if we can get rid of that, we don't know of any other potential important pathogens. I should say, one of the differences is that we've been living closely with pigs in our communities for many years, and yet we've not seen any outbreaks. We've also been working in xenotransplantation for 30 or 40 years, and none of the surgical staff have become sick. So that it's not that it could never happen, but I think the risk is modest. I think it's low. And so, but I think we should learn something. So I'm an advocate for metagenomic sequencing. We're looking undirected at the donors and the recipients to see if any new molecular species are present in donors and recipients under immune suppression. I've become an advocate for, before you put pig organs into people, immunosuppressing the pigs and seeing if anything new comes out. We may be surprised, but it's certainly an important experiment.
Thank you. Is there any other question from the audience? Alguma outra pergunta, gente, para... We know that the calcineurin inhibitors does not work like the for the immunosuppression, and most of the experiments are using co-stimulatory blockade. But do you think the mTOR inhibitors may have any function in terms of inhibiting viral replication?
So the mTOR inhibitors have been shown to have an advantage in terms of cytomegalovirus in particular, and because it does two things. It reduces production of early antigen, and it also seems to improve the T cell environment for fighting off viral infections in general. And so we know that that is advantageous in the respect of viral infections. The concern about the mTOR inhibitors, of course, is poor wound healing, and it causes proteinuria. So it probably would be disadvantageous for kidney xenotransplantation. It might be something that we can use later if somebody needs an additional agent. But I don't know that I would rely on that routinely.
I mean, in terms of pig CMV, do you know whether it works also on pig CMV?
No, don't know. And it's certainly worth studying, but I'd rather just not have the CMV in the graft, because even non-replicating CMV causes more intense graft rejection.
Yeah, thank you.
I have a follow-up question on the jump issue. So with the CRISPR technology, do you envision any way that you can shut down on some of these potentially harmful pathogens before transplant? Perhaps trying to avoid them to infect the recipient.
Yeah, I mean, it's an important consideration. I think in general, the only ones that you can knock out that will stay removed from the pigs are the endogenous retroviruses, so the ones that are in the genome. Otherwise, the exogenous viruses can reinfect the pig or the person later. And there's no question that the question would be, can human pathogens infect the pig organ? We always think about it as the pig infecting the human, but it would be a disaster if somehow it turned out that human organisms could infect pigs and disrupt pig production. So that I think the problem with CRISPR is that it can delete things that are in the genome and can actually make the cells resistant to reinfection of the retroviruses, but it's not going to be useful for other pathogens. The other pathogens, we have to exclude them from the colony or maybe develop vaccine strategies, or as we do in allotransplantation, prophylactic strategies with antiviral or antibacterial, antifungal agents.
Okay, any other question from the audience here? Let me check the chat.
I don't think
Não,
there
não
are
tinha nada,
questions
não.
in the chat.
Ninguém falou nada.
Yeah,
Pelo menos não tinha
there are
ninguém.
none. Anything else? Any comment?
Não sei se quando
No,
o
but if I
fala,
can
mas ele
help
ouviu também. Não
with
falou
your
Na
xenotransplant
3, sim.
studies, I'd be more than happy to, and thank you again for
Na
inviting me
3 e
back.
tem
I
um na
hope I can come
4
in
também.
person next time.
Na 4.
Thank you.
Thank you, Jake.
Me hand over
Na 3 é aquela
to Ed.
limítrofe.
I
Na
would
4,
like
deixa
just
eu
to
olhar.
thank you again. And as Jay remembered, he was here in our first course. Do you remember, Sylvia? And it's really a great pleasure. The efficient service at Mass General Hospital was a
Putz, eu fiz
model
tudo da
for
4,
us
não
to
vai
implement
aparecer agora.
our immunocompromised group here. We hope to receive you here as soon as possible. Okay, thank you again, Jay.
Look forward to it. Thank you all very much and have a great day and congratulations on your course.
Thank you.
Thank you, Jake. Bye. Good to see you.
Muito bem, então a gente agora abre para a discussão da mesa, né? Então eu queria convidar a Alice, Amanda e Edson para tomar assento aqui. Como atrasou, eu só peço para os 3 palestrantes, na hora que tiverem as perguntas, terem respostas curtas, para a gente não perder muito tempo. Então vamos começar. Silvia. Eu posso, pode deixar. Silvia é a passadora.
Então, gente, pergunta.
Eu tenho uma pergunta.
Elias.
A pergunta é para todos. A indústria farmacêutica, obviamente, só faz testes de drogas que lhes interessam vender. Então, são sempre monoterapias. Nós sabemos, por exemplo, dos estudos que vocês mostraram, inclusive dos estudos feitos pela doutora Lígia e pelo doutor Luiz Sérgio, no nosso serviço, que só 40% dos nossos pacientes desenvolvem anticorpos após a vacina. Nós sabemos que existem os antirretrovirais, nós sabemos que existem o soro convalescente. Mas nós nunca fizemos um estudo onde a gente pegue os pacientes imunossuprimidos, cuja mortalidade é 50% quando eles internam, e damos para esses pacientes, logo no início da infecção, um antirretroviral, um booster de vacinação e um soro de convalescente. Eu sei que custa caro, mas a indústria nunca vai fazer um estudo
Na 4 tem um Clostridium limítrofe positivo, na verdade, que é igual ao porte.
E a gente nunca usa
dia 25/10.
nem HIV,
Foi coletado
nem
ontem de novo.
nem nada. Então, a pergunta é por que a universidade não se salienta...
Não sei se ele não estava na 1, esse aqui antes, tá? Novo.
Só para lembrar que as autoridades talvez não saibam, o custo de uma diária hospitalar no nosso serviço é de R$ 1.800 por dia. Então, quando eles dizem que isso na unidade de internação, não na UTI,
Não, acho que ele está sempre na 4.
Custo efetivo, provavelmente porque nunca fizeram um estudo de custo-efetividade. Então, a minha pergunta é, não está na hora...
Mas não tem nenhuma banca prescrita, não.
Promover um estudo desse?
Não, acho que a Alice responde e, no final, a provocação do Elias, o Esper responde. Pode falar, Alice.
Oi, Elias. Então, os antivirais, né? Não antirretrovirais. Os antivirais têm poucos estudos com combinação. Até coloquei, não sei se você estava no último slide, talvez uma combinação de antiviral e antipopulacional à vista do que a gente tem de variantes possa ser mais efetivo, mas a gente não tem dados, né? Eu concordo que realmente o custo é muito alto, mas o custo, principalmente da população de transplante renal, né? Desde o começo do ano a gente tem visto um impacto significativo na mortalidade, mesmo em pacientes vacinados. É isso, e a gente chega, então, na política do hospital, né? Assim, de investir nos antivirais para uma terapia precoce, visando essa população de mais alto risco.
Esper quer complementar?
Não, eu acho que essa discussão tem seu lugar. O fato dessas pessoas estarem com viremia prolongada e, inclusive, desenvolvendo resistência, dá um déjà vu de outras viroses crônicas que a gente trata há muito tempo com combinações, como ele chamou atenção. Fazer esse estudo é factível, mas extremamente difícil, porque o número de pacientes que vão requerer esse tipo de intervenção vai ser muito pequeno, e quais combinações você vai usar, de que forma, sofre também o problema das modificações das subvariantes que vem acontecendo com o tempo, Inês. Então, por exemplo, se você tivesse desenhado, se nós dois estivéssemos sentados junto com o pessoal aqui da mesa, da Infecto, e bolado um estudo no começo do ano, a gente talvez fizesse uma combinação de anticorpos monoclonais com antiviral, etc. Hoje em dia, no meio do estudo, ia naufragar, porque as variantes desenvolveram resistência, a Evusheld desenvolveu resistência aos outros monoclonais da Eli Lilly, da Regeneron, etc. Então, é muito difícil. É claro que não é impossível, mas requeriria um acompanhamento e adaptação do estudo ao longo do tempo. Outra coisa também que a gente poderia ter sofrido, se a gente falar assim, ó, vamos fazer uma combinação de, digamos, remdesivir mais monopiravir mais plasma convalescente. Então, qual plasma convalescente? Se você pegar um plasma convalescente de 2021, ele já não funciona mais, porque as variantes são diferentes. Você teria que, de alguma forma, tentar também padronizar o plasma, mais uma variante no sistema, e torna isso mais difícil. Eu não acho que é impossível, mas seria um estudo com bastante variável, muito cêntrico, com um tamanho amostral muito grande para a gente conseguir tirar uma conclusão de desfecho significativo.
Eu queria só complementar um ponto que eu acho que concordo plenamente com a Alice e com essa. A Alice deixou clara essa dificuldade por causa das variantes, mas queria lembrar também que o nosso país ficou muito atrás em tudo que a gente poderia fazer para os nossos pacientes, né? Que até o momento a gente não tem disponível absolutamente nada, nem vacina bivalente no país. Então, parte do que a gente deveria estar fazendo do ponto de vista assistencial e acompanhando o que já está publicado na literatura, nós não conseguimos fazer, nem na medicina privada e nem no SUS. Então, estamos sempre 6 meses atrasados em relação à pandemia da Covid. Mais alguma pergunta? Tem uma pergunta aqui. Aí se apresenta, por favor.
Bom dia, primeiro quero agradecer por estar aqui hoje e parabenizar pela apresentação. Eu sou a Gabriela, sou MSL de Covid da MSD. E eu gostaria de saber a opinião de vocês com relação à representatividade da população de imunossuprimidos dentro dos estudos clínicos, como que vocês enxergam, visto que na maioria das vezes eles estão subrepresentados ou não representados, quão confiante vocês se sentem em extrapolar os dados dos estudos clínicos. E uma segunda pergunta também em relação a combinação que o Dr. Elias levantou, e eu também fiquei pensativa, qual seria a vantagem de combinar 2 antivirais para esses pacientes que foi apresentado ali?
Por exemplo, se o paciente
A vacina, eu disse até na minha aula, eles não fizeram parte dos estudos clínicos. Eu acho que a gente está extrapolando pelos estudos de vida real, né? Pelos estudos de efetividade, é isso.
Eu tenho paciente que já deu positivo
Não, com certeza estão sempre subrepresentados, né? Acho que até eu tinha a atenção do Evusheld, o número de imunodeprimidos, que é, na verdade, a nossa população-alvo a partir desse momento, né? Então, falta, assim, mas, assim, cada vez mais tem estudos com uma prevalência majoritária de imunossuprimidos, que é o que a gente quer. Agora, acho que precisa de estudos, né, para saber em relação à combinação de antivirais, qual seria esse impacto, se aumenta ali, se for ver naqueles estudos in vitro se separa, né, os da Gevimab e o Sisovimab, às vezes sozinhos eles não têm ação, mas em combinação tem uma melhor atividade. Acho que precisaria de estudos mesmo.
Desculpa, eu esqueci teu nome. Gabriela, assim, utilizar, extrapolar dados com pouca representatividade indefinidos já é a rotina de todo mundo, porque é sempre assim, e mesmo estudos específicos, algumas populações de imunossuprimidos, elas são maiores, mas naturalmente a população desconfrontada é uma população pequena, de avaliação difícil. Minha impressão, Alice me corrige, é que assim, à medida que os estudos são fortes, ainda que pouco representado, à medida que eles vão mostrando um benefício maior nessa população, acaba um pouco do que eu entendo, né? Assim, tentam se buscar. Não é que é uma população, no meu entendimento, pelo menos dos antivirais, de alguns estudos em Covid, não é que é uma população que ficou de fora dos trials, é uma população que nem sempre numericamente menor.
Também foi critério de exclusão, né? Então, quando chegavam os estudos, a gente tentava colocar os imunossuprimidos, colocava como critério de exclusão. E isso é um problema, viu? Porque muitas vezes esse critério de exclusão é também uma forma de você homogeneizar a amostragem, mas outra também do desenvolvedor da droga fugir de confusão. Aí, na hora que a gente chega, olha, vamos tentar, não, espera aí, vamos ver se vai dar certo, aprendendo competente.
Posso fazer uma pergunta?
A gente tem só mais uma pergunta e precisamos interromper para o COP. Então, acho que o Elias tem mais uma pergunta, né? É no chat.
Existem essas doenças induzidas por adjuvantes, e hoje a gente está vendo essa síndrome sendo mais identificada, que eles chamam de Asia ou Ásia Síndrome, que é autoimune inflammatory syndrome induced by adjuvants. Nas vacinações que nós tivemos, nós conseguimos identificar essa síndrome por adjuvantes vacinais?
Eu não tenho conhecimento dessa síndrome nas vacinas de Covid. Né? A gente teve reações específicas, inflamatórias, doenças autoinflamatórias identificadas, mas que eu saiba não foi atribuído ao adjuvante em si. Né? Essas doenças mais graves, como miocardite, a síndrome de trombose associada à plaquetopenia, não tem registro de que foi associado a adjuvante.
Obrigado.
Então, vou responder a última aqui que está no chat. O Hinaldo está perguntando em relação à utilização de teste e olhar CT para suspender isolamento. Eu estou fazendo questão de responder isso, porque isso entra naquele pacote de medidas que a gente... Ideal, embora o PCR positivo não signifique transmissão, CT podemos utilizar. Eu diria que depende do contexto, do tamanho que você tem. Eu vou dizer, gente, nós discutimos isso aqui algumas vezes. Em uma instituição que você faz o número de transplantes que faz, em que você tem o tanto de paciente oncológico que tem, Hinaldo, faz, no meu entendimento, faz um sentido científico, mas esse tipo de medida, a gente tem que tomar uma medida olhando a situação epidemiológica global isso eu tentei falar algumas vezes. fazer isso vai travando vários diversos processos assistenciais. Não sei, podemos discutir isso depois, se é o pensamento de todo. Então, há um sentido, mas provavelmente com um impacto que não é tão importante e acaba dificultando as demais ações ali. Querem encerrar? Podemos encerrar?
Encerramos então e temos um coffee, uma salva de palmas, né, para a mesa, que foi ótimo.
Um café para quem está online.
E o coffee aqui ao lado. Voltamos em
em
15 minutos,
15
gente.